Hi, my name is Cheng Liu, and I'm the director of antibody engineering, and I work for Trist Biopharma. So today I'm going to present you one of the library, human antibody library called Hyperimmune library. As you can see, this library actually is we made from mining of a human healthy donors or antibody sequence. And we used more than 2.5 million human HCDR3 sequence to construct this library. So Trist is a company. We are specialized in DNA synthesis and use Trist DNA synthesis technology, and we made high fidelity and high quality library for this specific antibody human library. So this library, actually, we only used single heavy chain framework and a single light chain framework. And we heavily included the diversity of HCDR3, but with limited HCDR1 and HCDR2 diversity. And you can see the total theoretical size is huge of this library. It's about 10 to the 20th, over 10 to the 20th. But the final library size we generated for this phage display library is 10 to the 10th. This is the distribution of the HCD of heavy chain. As you can see, the very limited heavy chain CDR1 distribution, length distribution, only one length for CDR1 and CDR2, but a very high even, high broad distribution for CDR3. This is fitting in a large human antibody library profile. This one shows us the Trist antibody discovery workflow. So once we had made this library, and we took us 2 weeks to do panning, either cell-based or protein-based selection. And then we take about 1 week, we do NGS and next generation sequencing to sequencing the selected final pools. And from the final pool, we reformatting all of the unique sequence. That takes about 4 weeks. And then we do high throughput IgG expression and purification, and use this purified antibody to do final binding and functional screening and assays. The whole process takes about 4 months. So for soluble protein selection, normally we use increased washing stringency, as you can see here. And then we use with decreased antigen present for the target. So this way we can increase the overall stringency of the selection. We normally do 5 rounds, but sometimes we actually only screen for round 3 or round 4. So here shows the general selection scheme as commonly used for everyone with the phage display. So we do washing and wash out the non-bind phage, and all of the remaining phage will be amplified and then be used for next round of selection. So here shows one of the soluble protein selection example. This one is a human TIGIT protein. As you can see, we only did 4 rounds of panning. We started with 100 picomolar concentration as the picomole as the antigen amount, and then it decrease antigen by 2-fold for each round of subsequent selection. As you can see, for both round 3 and round 4, we have identified many of the unique specific binders. This is the phage ELISA results, and after reformatting and purification of the protein, we identified here, we categorized 17 unique binders. So the affinity is about from 15 nanomolar to 300 nanomolar affinity. This is by the Birkhoff measurement. And you can see they are very diverse with different CDR lengths and also unique sequence, and also phylogeny. You can see the distribution and also affinity representation of this selected pool. So next example of the soluble selection, we use the CD3. As you know, it's very difficult to generate human and cyno CDR3 cross binders. So actually from this panning, 5 rounds panning, and we identified a huge number of binders. And from further analysis, you can see, and we identified one clone actually that binds both human CD3 epsilon and also cyno CD3 epsilon. So this is the top. This one is a human CD3 and cyno CD3, and this is one of the control murine SP34. As you can see, very weak binding affinity to both human and cyno CD3. And another clone shows they only bind a human CD3 with a 130 nanomolar affinity and almost no binding affinity to cyno CD3. Next on, we move on to demonstrate this library not only can be used for soluble protein and also can be for cell selection. So this one is for one of the GPCR target, CRTH2. So this is our selection scheme. So we express the GPCR protein with GFP at the C-terminal end and also flag tag at N-terminal end. So we select first with validated expression cells with both expression, both GFP expression and the flag tag expression. And here it shows one of the example, one of the clone we identified of the selection. They do binds to the expression cell line, and the black line is the parental cells, the parental CHO cells, and does not bind the parental CHO cells. And the bottom of this figure shows the CRTH2 CMP assay, and mostly our antibody shows the inhibitory activity of this CRTH2 activity. Here, not just one example, CRTH2 binders. We have here shows another 3 of CRTH2 with a different CMP inhibitory activity, and many of them with, all of them pretty much with sub-nanomolar IC50s. So here we shows this, we shows that we made a human antibody library. This library has been showed to bind select the binders for both TIGIT protein and also CD3 protein. And for the TIGIT target, we identified 17 unique antibody with the monovalent affinity from 16 nanomolar to over 300 nanomolar. And also for the CD3 target, we actually identified across binders, cross human and the cyno CD3 binders. And then next, we move on to demonstrate that we use this library to identify a panel of unique GPCR binding antibody, and also shows this antibody are fully functional, has a very potent inhibitory activity to the CRTH2 activation. So taken together, we demonstrate this is a high-quality hyperimmune library can be useful tool for antibody drug discovery.